Welcome, curious minds, to another deep dive. Today, we're not just scratching the surface, we're plunging microscopically deep into one of the most fundamental and actually quite intricate processes within your body. That's the metabolism of amino acids. Now, you might typically think of amino acids as just, the simple building blocks of protein, essential for growth, repair, all that. But as we're about to uncover, their breakdown, catabolism, is a far more complex and vital process than you might imagine. It has really profound implications for your overall health. This isn't just about what you eat, it's about how your body actually turns those nutrients into everything it needs.
Indeed. And for this deep dive, we'll be meticulously examining how the body handles the leftovers after nitrogen removal from proteins. Specifically, we're looking at the hydrocarbon skeletons of those protein amino acids. And like you said, this isn't merely academic biochemistry. Our journey today will illuminate the precise enzymes involved, the critical intermediate steps they catalyze, and maybe most importantly, what happens when things go awry. When pathways get disrupted, leading to what are often called inborn errors of metabolism. These conditions, while sometimes rare, they serve as powerful testaments to the absolute precision, the perfect orchestration required for our bodies to function optimally. It's a level of mastery that even elite professionals in the field strive to fully comprehend.
Right. So, our mission for you today is clear. We want to equip you with a truly master level understanding of this topic. Think of it as gaining the kind of insights that the elite professionals in this very field possess. We've sifted through, a dense stack of sources for this deep dive, but our goal is to extract only the most crucial insights. We want to reveal some surprising facts you might not expect and connect all the dots. By the end, you'll not only be well informed, but, dare I say, absolutely fascinated by the biochemical dance happening inside you every second. So let's unpack this intricate world together. All right, let's start at the very beginning with a seemingly simple question, but maybe it's not so simple. Why does our body even bother breaking down these amino acids in the first place, especially once the nitrogen has been stripped away? What's the ultimate destiny for these hydrocarbon skeletons? It almost seems, I don't know, counterintuitive.
That's an excellent question, actually. And it gets right to the heart of metabolic efficiency. The body doesn't waste resources. Excess amino acids, unlike, say, carbohydrates or fats, aren't simply stored as amino acids for later use. That's just not how our system is designed. Instead, once their amino groups have been efficiently removed, and we'll definitely get into how that happens shortly, the remaining carbon skeletons undergo catabolism. So they get broken down into what we call amphibolic intermediates. Now, amphibolic, that's a key term here because these molecules are truly fascinating. They sort of sit at the crossroads of multiple metabolic pathways. Gives the body incredible flexibility.
Amphibolic intermediates. Okay, that term itself suggests a lot of versatility, a lot of importance. So what exactly are they and why are they so central to metabolism? How do they provide that flexibility you mentioned?
Right, think of them as chemical compounds with a kind of dual destiny. They can be channeled in two primary metabolic directions. First, they can serve as a direct, immediate source of energy for the cell. This happens when they feed directly into energy-producing pathways, most notably the tricarboxylic acid cycle, or TCA cycle.
Krebs cycle.
Yeah, exactly, the Krebs cycle. Alternatively, and this is where their versatility really shines, they can be recycled. They can be used for the biosynthesis of other crucial macromolecules. We're talking about building new carbohydrates or even lipids, fats, depending on the body's needs at that moment. This remarkable interconvertibility of carbon atoms, where bits from fat, carbohydrate, and protein can all be repurposed, was a monumental discovery. It was established through, rigorous nutritional studies from the 1920s to the 1940s, and then powerfully reinforced by elegant studies using isotopically labeled amino acids, kind of the 40s and 50s. That early research really laid the foundation for understanding how efficiently our bodies manage energy and building blocks, constantly adapting.
That's incredibly efficient. So the body operates with almost no waste, constantly repurposing molecules based on what's needed right then and there. Could you give us a more precise sense of which amino acids turn into what, based on those categories you outlined? It feels like we're mapping out the body's internal recycling plant here.
Absolutely, and the sources categorize them quite clearly into three main groups based on the ultimate fate of their carbon skeleton. It reflects their ability to be channeled into these different metabolic pathways. So first, we have the glycogenic amino acids. These are primarily converted into precursors that can eventually form carbohydrates, glucose essentially. This usually means they become pyruvate or various intermediates of the TCA cycle. Think molecules like oxaloacetate or iketoglutarate or succinyl-CoA. This is a pretty extensive group. It includes alanine, arginine, aspartate, cysteine, glutamate, glycine, histidine, methionine, proline serine, threonine, and vainseline. And interestingly, it also includes 4-hydroxyproline, so even minor variations can influence that metabolic destiny. Then there are the ketogenic amino acids. These are converted exclusively to acetyl-CoA or acetoacetate. These end products are then typically used to form fats or ketone bodies, not glucose. This group is much smaller. It only includes leucine and lysine. Now finally, we have the most versatile ones, the glycogenic and ketogenic amino acids. These are the truly adaptable ones. They can form both carbohydrate and fat precursors. They have dual metabolic roles. This group includes isoleucine, phenylalanine, tryptophan, and tyrosine. So this broad categorization, it isn't just a list, right? It provides a comprehensive map. It shows how these essential building blocks are repurposed for energy and synthesis. Shows just how interconnected everything is.
That is a truly comprehensive map. It really illustrates the intricate planning within our biochemistry, how the body can constantly adapt. Now, okay, moving from these end products back to the very first step in this whole catabolic process, what's the crucial initial reaction for breaking down the vast majority of these amino acids?
Right. For the overwhelming majority of protein amino acids, the very first catabolic reaction is the efficient removal of the amino nitrogen. This absolutely critical process is called transamination, and it's catalyzed by a class of enzymes known as transaminases. You might also hear them called aminotransferases. These enzymes essentially perform a chemical swap. They transfer the amino group from the amino acid to a keto group from another molecule, typically an iketo acid like iketoglutarate or pyruvate. This produces a new iketo acid derived from the original amino acid and a new amino acid, often glutamate. Glutamate then serves as a kind of central collector of amino groups for eventual excretion, usually via the urea cycle. It's a reversible reaction, actually, which is crucial for redistributing nitrogen around the body, too.
So it's like a precise chemical dance-off where the amino group changes partners. Okay. And it sounds like a highly coordinated effort. Is there a specific coenzyme that's always involved in these transamination reactions? Does it act like a temporary partner or a facilitator?
Precisely. Aminotransferase or transaminase reactions invariably involve a specific and absolutely indispensable coenzyme, pyridoxal phosphate, PLP for short.
Ah, PLP.
Yeah, and this molecule is derived directly from vitamin B6, which highlights why getting enough B6 in your diet is so critical for healthy metabolism. PLP plays a pivotal role in facilitating the transfer of that amino group. It acts as a transient acceptor. It temporarily binds the amino group from the original amino acid, forms a short-lived intermediate called a Schiff base, and then efficiently transfers that amino group to the iketo acid acceptor. This allows the reaction to proceed smoothly, without sufficient vitamin B6.
Things grind to a halt.
Well, these crucial initial steps of amino acid catabolism would be severely impaired, yes. Could lead to a whole host of metabolic problems.
That's fascinating. So PLP from vitamin B6 acts like a vital shuttle. Okay. Given that most amino acids go through this transamination, are there any notable exceptions? Are there amino acids where their amino nitrogen is removed by entirely different processes?
Yes, there are indeed notable exceptions. And understanding them gives us deeper insight into the diversity of metabolic strategies the body uses. Proline, 4-hydroxyproline, threonine, and lysine. These four do not initiate their catabolism via traditional transamination. Their amino groups are handled, let's say, rearranged through entirely distinct, often more complex pathways. Once this initial nitrogen removal happens, whether by transamination or one of these alternative processes, the remaining hydrocarbon skeletons, those are then further degraded into those versatile amphibolic intermediates we talked about, ready to be used for energy or synthesis.
So what does all this intricate biochemistry mean for the human body if these crucial enzymes, whether the transaminases or the unique enzymes for the exceptions, what if they go awry? What are the real-world implications of these delicate pathways being disrupted?
This brings us to a very serious and medically critical aspect of amino acid metabolism. Disorders of amino acid catabolism, while comparatively rare in the general population, can have truly devastating consequences if they're left untreated. We are talking about conditions that can lead to irreversible brain damage, profound developmental delays, and even early mortality. This is precisely why prenatal or early postnatal detection, followed by timely initiation of the right treatment, is absolutely essential. It's quite literally life-saving for many affected individuals.
That sounds incredibly serious. Given the potential for such severe outcomes, how are these conditions typically detected? Are there widespread screening programs for newborns, or can they sometimes be identified even earlier?
Modern medicine has made truly remarkable strides in this area, especially in early detection. Prenatal diagnosis is often possible through procedures like amniocentesis. This allows for detection of specific enzyme activities in cultured amniotic fluid cells. gives parents and doctors a critical early warning. Postnatally, the screening efforts are even more widespread and comprehensive. In the United States, for example, all states conduct mandatory, comprehensive screening tests for newborns. These programs cover an impressive array, up to 40 different metabolic diseases. And this includes a significant number of disorders associated with defects in amino acid catabolism.
40 diseases, wow. That's an astonishing number to screen for. It must require incredibly precise and efficient technology to be practical at a population level. What's the most reliable method they use now to screen for these conditions, particularly in newborns?
You're absolutely right. Efficiency and reliability are paramount. Given the sheer number of conditions and the critical time frame for intervention, the most reliable and widely adopted screening tests currently utilize tandem mass spectrometry, MSMS.
Ah, mass spec.
Exactly. This technology is truly a game changer in newborn screening. With just a few drops of a neonate's blood, usually collected on a special filter paper card shortly after birth, this sophisticated technology can accurately detect and quantify a wide range of catabolites. Those are the specific breakdown products, catabolites that are indicative of a particular metabolic defect. By identifying these accumulating catabolites, the test can then implicate the absence or significantly lowered activity of one or more specific enzymes in that pathway. This powerful tool has revolutionized early detection. It allows for crucial interventions before irreversible damage occurs, fundamentally changing the prognosis for many of these conditions. Now, at the heart of all these metabolic disorders are fundamental genetic mutations. A mutation, whether it occurs directly within the gene that encodes a specific enzyme, or even in the associated regulatory regions of DNA that control when and how that gene is expressed, it can lead to several different problems impacting the enzyme itself. It could result in the complete failure to synthesize the enzyme, meaning just isn't made at all. Alternatively, it might lead to the synthesis of an enzyme that is only partially functional, maybe working at reduced efficiency or it's unstable. Or it could result in the synthesis of an enzyme that is completely non-functional, effectively useless to the cell, even though it might be present.
So it's not just about an enzyme being absent. It can be there, but just not working right, or working at, say, a fraction of its capacity. That adds a remarkable layer of complexity to understanding these conditions. What specific ways can these mutations actually impact an enzyme's function?
Exactly. These mutations can affect enzyme activity in myriad, sometimes subtle ways. For instance, a mutation might compromise the enzyme's crucial three-dimensional structure, which is absolutely critical for it to bind its substrate and catalyze their reaction. It could disrupt its active site, the specific region where the chemical reaction takes place, or its regulatory sites, where other molecules bind to modulate its activity. Mutations can also impair the precise positioning of critical amino acid residues, the ones directly involved in catalysis or in binding the substrate, or maybe its essential coenzyme like PLP, or even a necessary metal ion. Furthermore, mutations might hinder an enzyme's ability to respond appropriately to allosteric regulators.
Those are the molecules that bind elsewhere but still affect activity.
Precisely. They bind at a site distinct from the active site, but still influence the enzyme's overall activity, either activating or inhibiting it. A mutation could mess that up.
Okay, here's where it gets really interesting, and it's a point our sources emphasize that really sticks with you. Different mutations can apparently give rise to the same clinical signs and symptoms in a patient, even if the underlying molecular defect is distinct. How does that happen?
That's a crucial point in understanding the clinical presentation of these diseases. It's quite counterintuitive at first. While various mutations may indeed give rise to the same observed clinical signs and symptoms in a patient, so what you see and measure might look strikingly similar. At a molecular level, these can represent distinct molecular diseases. This occurs because different mutations, even if they're in different parts of the same gene, or sometimes even in different genes within a related pathway, they can ultimately have similar downstream effects. For example, several different mutations might all severely compromise the enzyme's catalytic efficiency, or they might all lead to a similar disruption in a critical regulatory response. The net result for the patient can appear identical, even if the precise molecular cause is unique. This heterogeneity makes diagnosis, and sometimes treatment, a fascinating challenge.
And once these metabolic disorders are detected, especially given the serious consequences if left untreated, what's the primary way they are managed?
Primarily, the most common and effective remediation for these disorders of amino acid catabolism consists of feeding patients diets that are specifically low in the particular amino acid whose catabolism is impaired. The fundamental goal here is to reduce the buildup of the toxic intermediates that accumulate when the enzyme responsible for their breakdown is deficient or non-functional.
Right, limit the input.
Exactly. By carefully limiting the intake of the problematic amino acid, you effectively limit the production of these harmful substances, thereby mitigating their toxic effects. However, the ultimate hope, which is still largely, a future frontier, is genetic engineering. To permanently correct the underlying metabolic defect, this would involve directly altering the faulty gene to restore normal enzyme function, offering a more definitive and potentially curative solution, moving beyond just managing symptoms through diet, #tag dissecting specific amino acid catabolic pathways and disorders.
All right, let's begin our deep dive into the specific amino acid pathways themselves. We'll start with what are perhaps some of the more, let's say, straightforward ones, relatively speaking, in this complex system. What happens to asparagine and aspartate once they enter the body's catabolic machinery?
For asparagine and aspartate, the pathways are indeed relatively short and direct. They lead straight to central amphibolic intermediates. All four carbons of both asparagine and aspartate ultimately form oxaloacetate.
A key TCA cycle player.
Exactly. Right in the heart of the TCA cycle. The breakdown begins with asparaginase. That's an enzyme that converts asparagine to aspartate by hydrolyzing its amide group. Once you have aspartate, a transaminase enzyme then catalyzes its conversion to oxaloacetate, typically by transferring its amino group to an aceto acid like acetoglutarate, forming glutamate in the process. Pretty neat.
And glutamine and glutamate. Is their metabolic fate similar to asparagine and aspartate in terms of, well, simplicity and end product?
Similarly, yes. The catabolism of glutamine and glutamate also leads directly to a central TCA cycle intermediate. Successive reactions, first catalyzed by glutaminase, which converts glutamine to glutamate by removing its amide nitrogen, and then by a transaminase, convert glutamate into ketoglutarate, another crucial intermediate of the TCA cycle. Again, this transamination often involves transferring the amino group to pyruvate to form alanine, or maybe to oxaloacetate to form aspartate. What's truly fascinating here, and a core insight into why we don't see certain metabolic diseases, is this. While both glutamate and aspartate are substrates for these very same transaminases, metabolic defects in these specific transaminases, the ones that fulfill these absolute central amphibolic functions, are often incompatible with life.
They're just too critical.
Precisely. These enzymes are so fundamentally critical, they have such broad, ubiquitous roles in metabolism, that a significant deficiency in their activity would likely be lethal, probably during embryonic development or very early in life. Consequently, there are no known viable metabolic defects associated with these two very short but profoundly important catabolic pathways. Their importance means any severe impairment is likely fatal before birth or in the earliest stages, which prevents clinical observation of those specific defects.
That makes perfect sense. If something is so central, its disruption would be catastrophic. Okay. You mentioned earlier that proline doesn't undergo that initial transamination reaction like most amino acids, which immediately makes its pathway distinct. How exactly is its amino nitrogen handled during its catabolism?
That's right. Proline's pathway is one of those crucial exceptions. Its catabolism takes place entirely within the mitochondria.
Inside the mitochondria, okay.
Yeah, emphasizes the compartmentalization of metabolism. What's unique is that its amino nitrogen is actually retained throughout a two-stage oxidation process that ultimately forms glutamate. The first oxidation step is catalyzed by an enzyme called proline dehydrogenase. This enzyme converts proline into O1-pyrroline 5 carboxylate. Following that, the subsequent oxidation to glutamate is catalyzed by another distinct enzyme, O1-pyrroline 5 carboxylate dehydrogenase. This one's also known as glutamate semialdehyde dehydrogenase. So you see, the nitrogen isn't removed in an initial transamination step. Rather, it's incorporated into the glutamate structure through these specific oxidative steps within the mitochondria.
So two distinct mitochondrial enzymes are key here. Proline dehydrogenase and O1-pyrroline 5 carboxylate dehydrogenase. Are there disorders linked to deficiencies in these specific enzymes? And if so, what are their characteristics?
Yes, there are two distinct metabolic disorders of proline catabolism. And interestingly, both are inherited as autosomal recessive traits. Surprisingly, unlike many other amino acid disorders, these are generally consistent with a normal adult life, which tells us something about the relative tolerance of the body to their disruption compared to, say, defects in the TCA cycle itself. The first is type I hyperprolinemia. In this condition, the metabolic block occurs specifically at the enzyme proline dehydrogenase. A key diagnostic point here is that this defect does not impair the catabolism of 4-hydroxyproline. It shows the specificity of this enzyme for proline. Individuals with this condition typically have elevated proline levels, but are often asymptomatic or maybe have mild, nonspecific symptoms. The second is type 2 hyperprolinemia. In this case, the metabolic block is at 1-pyrroline 5-carboxylate dehydrogenase. Now, this enzyme is crucial because it also participates in the catabolism of arginine, ornithine, and 4-hydroxyproline.
Ah, so it hits multiple pathways.
Exactly. Because both proline and 4-hydroxyproline catabolism are affected by this particular defect, you'll see the characteristic excretion of both a 1-pyrroline 5-carboxylate from choline and 1-pyrroline 3-hydroxy5-carboxylate, 4-hydroxyproline in the urine. This dual excretion is a key diagnostic indicator for type 2. And while often benign, severe cases can sometimes present with neurological symptoms.
What about 4-hydroxyproline specifically? You just mentioned it gets affected in type 2 hyperprolinemia, but does it have its own dedicated catabolic pathway and associated disorders independent of proline?
Yes, 4-hydroxyalproline catabolism is indeed a multi-step process. It eventually yields glyoxylate and pyruvate, often through an aldol-type cleavage reaction. Just as with proline, there's a specific defect unique to its pathway. A deficiency in the enzyme 4-hydroxyproline dehydrogenase results in hyperhydroxyprolinemia. This condition is generally considered benign, meaning it typically doesn't cause severe health problems. And importantly, it does not affect proline catabolism. However, as we discussed with type 2 hyperprolinemia, a defect in glutamate semialdehyde dehydrogenase, that common enzyme involved in the second step of proline catabolism, that is accompanied by the excretion of A1-pyrroline 3-hydroxy5-carboxylate. This highlights how these seemingly distinct pathways can converge at certain enzymatic steps, leads to interconnected clinical presentations, and offers valuable diagnostic clues. Okay, moving on to arginine and ornithine. Their catabolic pathways are intricately connected with the body's central nitrogen disposal system.
The urea cycle, right?
Yes, exactly, the urea cycle. The initial reactions in arginine catabolism involve its conversion to ornithine. This conversion is catalyzed by arginase, a key enzyme that is part of the urea cycle itself. Plays a vital role in detoxifying ammonia. Once ornithine is formed, it is then transaminated to glutamate semialdehyde. From there, glutamate semialdehyde proceeds to be further catabolized to aketoglutarate, very similar to how it occurs in proline's breakdown pathway, as we just discussed. So you see, the body often recycles intermediate steps for efficiency, saves energy and resources.
So these pathways are indeed deeply intertwined with nitrogen disposal, making them crucial for overall metabolic health. What specific disorders show up when there are mutations or defects in the enzymes involved in arginine and ornithine catabolism?
There are two notable disorders associated with this pathway, both highlighting the importance of the urea cycle and related amino acid metabolism. First, mutations in the enzyme ornithine aminotransferase, also called ornithine transaminase, lead to elevated plasma and urinary ornithine levels. These elevated levels are specifically associated with a progressive eye disease called gyrate atrophy of the choroid and retina.
Gyrate atrophy.
Yes, it results in vision loss due to degeneration of the light-sensitive tissues at the back of the eye. The primary treatment strategy for this condition involves restricting dietary arginine. Since arginine is the direct precursor to ornithine, this reduces the buildup of the toxic ornithine levels. The second significant condition is the hyperornithinemia hyperammonemia syndrome, often abbreviated as HH8 syndrome.
HHA.
Okay. This complex disorder is caused by a defective ORC1 mitochondrial ornithine citrulline antiporter. This antiporter is a critical transporter protein. It's responsible for moving ornithine from the cytoplasm into the mitochondria, where it plays a crucial role in the urea cycle's efficient functioning. When this transporter is defective, ornithine cannot enter the mitochondria efficiently. This leads to a disruption of urea synthesis and a subsequent accumulation of toxic ammonia.
Hyperammonemia.
Exactly, hyperammonemia. In addition to the hyperornithinemia, this can lead to severe neurological symptoms. It's quite serious.
Moving on to histidine. How does its breakdown pathway offer a fascinating and surprisingly clever diagnostic clue for other nutritional deficiencies, specifically folic acid deficiency?
Right, histidine catabolism is a multi-step process that proceeds through several distinct intermediates. These include urocanate, then 4-imidazolone 5-propionate, and then N-formimino-glutamate, often abbreviated as FIGLU.
FIGLU. Got it.
The crucial step where this diagnostic connection arises is the transfer of the formimino group from FIGLU to tetrahydrofolate. This reaction is absolutely essential and forms glutamate, which then subsequently becomes ketoglutarate, feeding into the TCA cycle. Now, here's the diagnostic connection, and it's quite elegant. In the case of folic acid deficiency, the body's ability to transfer this formimino group from FIGLU to tetrahydrofolate is severely impaired. Why?
because tetrahydrofolate itself is in short supply.
Precisely. As a direct consequence, Phy-glue cannot be processed efficiently and thus accumulates. This leads to its excessive excretion in the urine. Therefore, measuring Phy-glue excretion in a patient's urine, particularly after they've been given a standard dose of histidine to sort of challenge the pathway, that can be used as a reliable and clever diagnostic tool to detect an underlying folic acid deficiency. It's a metabolic bottleneck revealing a broader nutritional issue. Very neat.
That's a truly ingenious diagnostic trick, using a specific metabolic bottleneck to reveal a broader nutritional issue. Are there any other disorders related directly to histidine catabolism itself?
Yes, there are other generally benign disorders of histidine metabolism. It's important to distinguish these from more severe conditions. These include histidinemia. This is due to impaired activity of the enzyme histidase, the very first enzyme in the histidine degradation pathway. Results in elevated histidine levels in blood and urine. Another condition is urocanic aciduria. This is associated with impaired urokinase, the enzyme that acts on urokinase, the second intermediate. Both of these conditions are typically not associated with severe clinical consequences, unlike many of the other amino acid disorders we've discussed. It highlights that not all metabolic defects lead to severe pathology. Some are relatively mild.
Let's group some of these smaller amino acids, glycine, serine, alanine, cysteine, and cysteine, as their transformations can be quite diverse and reveal different metabolic strategies. How does glycine in particular get broken down? It's a very simple amino acid structurally, but its pathway seems to be anything but simple.
Glycine catabolism is indeed unique. It doesn't follow the common transamination route. It's handled by what's called the glycine cleavage system. This operates specifically in the liver mitochondria. This system is remarkable because it directly splits glycine into carbon dioxide, CO2, and ammonium, NH4+. While doing that, it also forms N5000N10 methylene tetrahydrofolate, a crucial one-carbon unit donor used in many biosynthetic reactions.
So it's not just breaking it down, it's producing something useful too.
Exactly, and this isn't a single enzyme, but a complex enzymatic system. It consists of 3 distinct enzymes, sometimes called P, T, and L proteins, and a unique H protein that has a covalently attached dihydrolipoyl moiety, which is essential for the reaction sequence. So it's a direct oxidative decarboxylation and deamination, a very efficient way to completely degrade this small amino acid while capturing a key building block.
What happens when that intricate glycine cleavage system isn't working correctly? Or if other parts of glycine metabolism are impaired, are there significant disorders here?
Oh yes. When this system is defective, it can lead to very serious consequences. A rare but typically severe inborn error is non-ketotic hyperglycemia. This results from a defect in glycine degradation by this cleavage system. It causes glycine to accumulate to toxic levels in all body tissues, particularly the central nervous system, leads to profound neurological problems, severe developmental delay, seizures, hypotonia, often fatal early in life. Another significant disorder is primary hyperoxaluria. This condition is characterized by a failure to properly catabolize glycolate, which is a compound formed from the deamination of glycine. When glycolate cannot be processed, it is subsequently oxidized to oxalate.
And oxalate is bad news for kidneys, right?
Very bad news. The accumulation of oxalate leads to the formation of painful kidney stones, that's urolithiasis, and calcification of the kidneys themselves, nephrocalcinosis. This can ultimately cause early mortality from renal failure or hypertension. It's a very serious condition. There's also glycinuria. This is a defect resulting from impaired renal tubular reabsorption of glycine, causing its excessive excretion in urine. This one is usually more benign, though.
So serine also has a close connection to glycine, right? Does its catabolism eventually feed into the glycine pathway, linking these two?
Yes, that's correct. Serine catabolism indeed merges with that of glycine, making their fates interconnected. This occurs after serine is converted directly to glycine. This reaction is catalyzed by the enzyme glycine hydroxymethyltransferase. So once serine becomes glycine, its further breakdown, including the removal of its carbon and nitrogen, follows that unique and critical glycine cleavage system pathway we just discussed.
What about alanine? It's another relatively simple amino acid, and we've seen it pop up as an end product in some transamination reactions.
Alanine is a more straightforward case among these smaller amino acids. It undergoes a simple transamination reaction, which we discussed as the most common initial step for many amino acids, to form pyruvate.
Right, pyruvate, the metabolic crossroads.
Exactly. Pyruvate can either enter the TCA cycle for energy generation or be used for glucose synthesis via gluconeogenesis. Interestingly, likely due to its central and ubiquitous role in metabolism, literally sitting at the nexus of carbohydrate and amino acid metabolism, there is no known viable metabolic defect of alanine catabolism. A deficiency in such a fundamental enzyme would probably be incompatible with life, again underscoring the vital importance of certain metabolic steps.
In cysteine, how does its breakdown proceed, and what are the implications for the diverse disorders we see related to its metabolism? It seems quite complex.
Cysteine metabolism indeed involves a diverse set of pathways and associated disorders. It highlights its complex role and susceptibility to disruption. It often begins with a preliminary step for its oxidized dimer form, cysteine. Cysteine is first reduced back to cysteine by the enzyme cysteine reductase. Once in the cysteine form, it can then be converted to pyruvate via two different catabolic pathways. Both involve oxidation and desulfuration steps. And yes, there are numerous abnormalities associated with cysteine metabolism. One prominent disorder is cysteine lysinuria, more commonly known as cystinuria. Now, this is not an enzymatic defect, interestingly. It's a defect in the renal tubular reabsorption of not only cysteine, but also lysine, arginine, and ornithine. These structurally related amino acids share a transporter.
So it's a transport problem, not a breakdown problem.
Correct. This leads to their excessive excretion in the urine. Apart from the potential for the formation of cysteine calculi, or kidney stones, which can be quite painful and problematic due to cysteine's low solubility, this condition is generally considered benign. It's managed largely by increasing fluid intake and sometimes alkalinizing the urine to help keep cysteine dissolved. A fascinating detail for patients with cystinuria: they often excrete a mixed disulfide of L-cysteine and homocysteine. This mixed disulfide is actually more soluble than pure cysteine, which fortunately helps to reduce the formation of those problematic cysteine kidney stones, a small silver lining perhaps. Then we have the homocysteinurias. This is a group of disorders. They can manifest as either vitamin B6 responsive or vitamin B6 unresponsive forms, which indicates different underlying enzymatic issues. A common cause is the deficiency of the enzyme cystathionine desynthase. This enzyme catalyzes the crucial reaction combining serine and homocysteine to form cystathionine.
And that enzyme needs B6.
Exactly. It requires PLP, the active form of B6, as a coenzyme. This explains the B6 responsive forms. Giving extra B6 can sometimes boost the residual enzyme activity. The consequences of homocysteinurias can be severe, including osteoporosis, dislocation of the ocular lens, which is called ectopia lentis, and often intellectual disability. It's also worth noting, while epidemiological and other data link elevated plasma homocysteine levels to increase cardiovascular risk, its direct role as a causal cardiovascular risk factor remains a subject of ongoing scientific controversy. It's complex to prove direct causation in human disease. Another significant condition is cystinosis, or cysteine storage disease. This is due to a defective carrier-mediated transport of cysteine, specifically out of lysosomes.
Lysosomes,
Yeah.
the cell's recycling centers.
Precisely. When this transporter is defective, cysteine accumulates as insoluble crystals within various tissues throughout the body: kidneys, eyes, liver, spleen. This ultimately leads to organ damage and early mortality, often for acute renal failure. It highlights the importance of proper intracellular transport and cellular waste disposal.
And what about threonine? How does its catabolism proceed? Does it connect to any of the pathways we've already discussed?
Threonine's catabolism involves an interesting cleavage reaction. The enzyme threonine aldolase cleaves threonine into two smaller molecules, glycine and acetaldehyde.
Ah, so it feeds into the glycine pathway.
It does. As we discussed, the catabolism of glycine then proceeds through the glycine cleavage system, ultimately leading to CO2 and NH4 plus styrene. The acetaldehyde produced is subsequently oxidized to acetate by aldehyde dehydrogenase, and acetate then forms acetyl-CoA, which can readily enter the TCA cycle or be used for fatty acid synthesis. So threonine ultimately contributes to both glycogenic via glycine and ketogenic via acetyl-CoA pools, showcasing its versatile metabolic fate.
Now let's tackle the aromatic amino acids. These tend to have more complex structures and, consequently, more intricate pathways. We'll start with tyrosine. Its degradation pathway seems to have many steps.
Tyrosine catabolism is indeed quite involved, and it uniquely results in both glycogenic and ketogenic end products, making it a versatile fuel source. It begins with the transamination of tyrosine to pi-hydroxyphenyl pyruvate. From there, it goes through a series of successive reactions catalyzed by several key enzymes. These include enzymes like pi-hydroxyphenyl pyruvate hydroxylase, homogentisate oxidase, and fumarylase acetoacetate hydrolase. Ultimately, the carbon skeleton of tyrosine is elegantly degraded into two distinct amphibolic intermediates, fumarate and acetoacetate.
Fumarate for the TCA cycle, acetoacetate for ketones or fats.
Exactly. Fumarate is a direct TCA cycle intermediate making tyrosine glycogenic. Acetoacetate can readily yield acetyl-CoA and acetate, making it ketogenic. This dual nature makes tyrosine a valuable energy and building block source.
Given how many steps there are in the breakdown of tyrosine, I imagine there are several associated disorders that can arise if any of these enzymes are defective. This seems like a pathway particularly vulnerable to genetic errors.
You're absolutely right. There are several clinically significant and distinct metabolic disorders associated with the tyrosine catabolic pathway, each resulting from a defect in a specific enzyme along the way. First, we have type I tyrosinemia, also known as tyrosinosis. This is generally the most severe form. The probable metabolic defect here is that the enzyme fumarate acid ketoacetate hydrolase, that's the last enzyme in the pathway. If left untreated, both acute and chronic forms of tyrosinosis typically lead to severe liver failure and kidney tubular dysfunction, often resulting in early death. Therapy primarily involves a strict diet low in both tyrosine and phenylalanine.
Why phenylalanine too?
Because phenylalanine is converted to tyrosine. So limiting phenylalanine intake also helps reduce the load on the defective pathway downstream. Next is type 2 tyrosinemia, also called Rittner-Hanhart syndrome. This disorder involves a defect in tyrosine aminotransferase. That's the very first enzyme in the tyrosine catabolic pathway. This deficiency leads to accumulation of tyrosine itself and the excretion of alternate tyrosine metabolites. It classically affects the eyes, causing corneal lesions, the skin, causing painful hyperkeratotic lesions, and sometimes causes intellectual disability. Then there is neonatal tyrosinemia. This is often a more transient condition. It's usually due to lowered activity of hydroxyphenyl pyruvate hydroxylase. This enzyme's activity could just be immature in newborns, especially premature ones. The therapy for this condition typically employs a diet low in total protein, rather than just specific amino acids. And it often resolves as the enzyme matures over weeks or months. Finally, we have alkaptonuria. This is a historically fascinating disorder. It's caused by a defective homogentisate oxidase. This enzyme normally catalyzes the breakdown of homogentisate, an intermediate in the tyrosine pathway. The classic and most striking sign of alkaptonuria is urine that darkens significantly on exposure to air.
Right, that's a famous one.
Exactly. This darkening occurs due to the oxidation of the excreted homogentisate when it reacts with oxygen. In the late stages of the disease, it leads to severe arthritis, particularly in large joints and the spine, and a distinctive bluish-black connective tissue pigmentation known as ochranosis. This ochranosis occurs because the accumulating homogentisate is oxidized to benzoquinone acetate, which then polymerizes and binds irreversibly to collagen and connective tissues throughout the body, causes the characteristic dark discoloration, and contributes to the joint damage.
That darkening urine sounds like something straight out of a medical mystery novel. It's such a unique and observable symptom.
It truly is, and alkaptonuria was first described as early as the 16th century, precisely based on that striking observation. But its significance goes far beyond its curious symptom. Alkaptonuria actually provided the fundamental basis for Sir Archibald Garrod's classic ideas in the early 20th century.
About inborn errors of metabolism.
Precisely. What he termed inborn errors of metabolism. He was one of the first to recognize that specific enzyme deficiencies could lead to specific disease phenotypes, a groundbreaking concept. And in a truly remarkable twist of historical and scientific discovery, based on the presence of ochranosis in skeletal remains and chemical evidence, the earliest known case of alkaptonuria was detected in 1977 in an Egyptian mummy dating from 1500 BC.
No way, 1500 BC.
Yes. It demonstrates that it is a truly ancient disease, yet our modern diagnostic insights allow us to identify it across millennia, connecting deep history with contemporary biochemistry.
Wow, an Egyptian mummy. That's a deep dive into history right there, connecting ancient pathology with modern biochemistry. Incredible. So phenylalanine also fits into this category of aromatic amino acids with complex catabolism. What's its story, and what are the most significant disorders associated with its breakdown, especially given its dietary importance and, well, PKU?
Phenylalane's catabolism is intimately linked with tyrosine, because for it to be broken down, phenylalanine is first converted to tyrosine. This crucial initial reaction is catalyzed by the enzyme phenylalanine hydroxylase. After this vital conversion, its catabolism then follows precisely the same reactions as tyrosine, leads to fumarate and acetoacetate, making it both glycogenic and ketogenic. The most well-known and clinically significant disorders related to phenylalanine metabolism are collectively called the hyperphenylalanemias. The most common and clinically significant is type I hyperphenylalaninemia, universally known as classic phenylketonuria, or PKU.
PKU.
This condition occurs in about 1 in 10,000 births. It's due to a defect specifically in the enzyme phenylalanine hydroxylase. This deficiency prevents the proper conversion of phenylalanine to tyrosine, leading to a toxic buildup of phenylalanine in the body. Other types of hyperphenylalaninemia, specifically types 2 through V, are less common but equally important. These arise from defects not in the hydroxylase enzyme itself, but in the metabolism of dihydrobiopterin, or BH4.
The coenzyme.
Exactly. This coenzyme is absolutely essential for the proper activity of phenylalanine hydroxylase. So these defects can be either in dihydrobiopterin reductase, which regenerates BH4, or in BH4 biosynthesis itself. In all these cases, regardless of the specific enzymatic defect, the common outcome is that toxic accumulation of phenylalanine. This excess phenylalanine then gets shunted into alternative catabolic pathways, leads to the excretion of abnormal alternative catabolites in the urine, such as phenylpyruvate. The truly good news here, and really a triumph of modern medicine and biochemistry, is that a strict diet low in phenylalanine, initiated early in life, can effectively prevent the severe mental retardation and neurological damage associated with classic PKU. This emphasizes the profound importance of early diagnosis and intervention. It's a condition where treatment can literally change a life's trajectory.
That's a powerful example of how crucial early detection is. How is PKU typically diagnosed in newborns, especially given its prevalence and the absolute need for immediate intervention?
Right, for prenatal diagnosis, DNA probes are now available. They can facilitate the detection of defects in phenylalanine hydroxylase or dihydrobiopterin reductase, offering insights before birth if there's a family history. For newborns, where mass screening is vital, there are some important considerations. Elevated blood phenylalanine levels might not be detectable until 3 to 4 days postpartum. The infant needs to have consumed protein for phenylalanine to accumulate to measurable levels. Also, false positives can occasionally occur in premature infants due to the delayed maturation of the enzymes involved. Historically, an older and frankly less reliable screening test involved using ferric chloride, FeCl3, to detect urinary phenylpyruvate, one of those alternative catabolites.
Yeah, color change test.
Yeah, if present, it would react with the FeCl3 to tune the urine a distinctive green-blue color. While FeCl3 screening for PKU in newborn urine was compulsory in many countries for decades, it has largely been supplanted in the United States and many other places by the much more reliable and comprehensive tandem mass spectrometry. MSMS can detect multiple metabolic disorders simultaneously and with far greater accuracy, further improving early intervention rates and reducing false positives. It's really the gold standard now for newborn screening.
Lysine was another amino acid you highlighted as an exception to the general transamination rule for nitrogen removal. That really piqued my interest. How is its nitrogen handled? And what are the fascinating intricacies of its unique catabolic pathway?
You're right, lysine's pathway is particularly unique because neither of its nitrogen atoms, not the amino nitrogen, nor the nitrogen on the side chain, participates in transamination in the traditional sense, unlike most other amino acids. Instead, the nitrogen of lysine is removed initially through a complex series of steps, starting with the formation of a compound called saccharapine. Subsequent reactions in the pathway then liberate the nitrogen as well. The ultimate product of the carbon skeleton breakdown is crotynyl-CoA. This complex pathway involves a truly remarkable bifunctional enzyme called amino-adipate semialdehyde synthase.
Bifunctional, so it does two jobs.
Exactly. This single enzyme actually possesses two distinct enzymatic activities on its different domains. It has a lysine-ketoglutarate reductase activity on its N-terminal part and a saccharapine dehydrogenase activity on its C-terminal part. Very efficient design. The pathway progresses through several key intermediates, including li-amino-adipate semialdehyde, then li-amino-adipate, followed by aketoadipate. This then leads to the formation of glutaryl-CoA, which is subsequently decarboxylated to crotynyl-CoA. Finally, crotynyl-CoA is reduced by crotynyl-CoA reductase to form butynyl-CoA, and from there, the reactions are entirely analogous to those found in fatty acid catabolism.
Wow, so it feeds right into fat breakdown pathways at the end.
Demonstrates a clever and efficient convergence of metabolic pathways, yeah.
And what about the related disorders that can arise from defects in this intricate lysine catabolic pathway? It sounds like there are many points where things could potentially go wrong.
There are indeed several significant disorders linked to lysine catabolism. Hyperlysinemia can result from a metabolic defect in either the first or the second activity of that remarkable bifunctional enzyme we just discussed, amino-adipate semialdehyde synthase. However, it's important to note that only if the defect involves the second activity, the saccharapine dehydrogenase activity, is it accompanied by elevated levels of saccharapine in the blood.
So detecting saccharapine helps pinpoint the exact defect.
It does. It's a key diagnostic marker for that specific subtype. There's also a distinct inherited metabolic disease that results from a defect later in the pathway, specifically at the decarboxylation of glutyl-CoA to crotonal-CoA. This condition is associated with severe striatal and cortical degeneration within the brain, leads to significant neurological symptoms, and is characterized by significantly elevated concentrations of glutarate and its related metabolites, like glucoconate and 3-hydroxyglutarate, in urine and blood. The clinical challenge in managing these metabolic defects is quite significant. It requires carefully restricting the dietary intake of lysine to prevent the accumulation of toxic intermediates, but doing so without causing malnutrition, which is a delicate and difficult balance to achieve, especially in growing children who absolutely require lysine for protein synthesis. Tryptophan, well known for its role in producing neurotransmitters like serotonin and the sleep hormone melatonin, but it also has a catabolic pathway for its breakdown into energy-yielding compounds, right? How does that proceed, and what makes it unique? Absolutely. Tryptophan is a truly fascinating amino acid with multiple crucial metabolic fates. Its catabolism, leading to amphibolic intermediates, occurs primarily via what's known as the kynurenine and thranolate pathway. The very first and usually rate-limiting enzyme in this pathway is tryptophan 2-color-3-dioxygenase, also called tryptophan pyrrolase. This enzyme initiates the breakdown by opening the indole ring of tryptophan, incorporating molecular oxygen, and forming N-formylkynurenine. This enzyme itself is quite interesting from a regulatory perspective. It's an iron porphyrin metalloprotein.
Meaning it has iron, like hemoglobin.
Sort of, yeah. It contains iron within a heme-like porphyrin ring. Its activity in the liver is inducible by adrenal corticosteroids and also by tryptophan itself. That indicates a feedback loop where increased tryptophan leads to more enzyme to break it down. Makes sense. Furthermore, it is feedback inhibited by nicotinic acid derivatives, including NADPH, suggests a link to the body's energy status and NAD+ production. After N-formylkynurenine is formed, the formal group is then hydrolytically removed by an enzyme called kynurenine formulase, producing kynurenine. A subsequent enzyme, kynureninase, importantly requires pyridoxal phosphate, PLP, our familiar vitamin B6 derivative again.
B6 pops up
It
everywhere.
really does. This PLP requirement is key for diagnostic insights here too.
That's a lot of intricate steps and regulatory controls. Any disorders here that give us unique insights, perhaps beyond just tryptophan metabolism itself, maybe revealing other nutritional deficiencies?
Yes, there are two particularly insightful conditions related to tryptophan catabolism. First, that requirement of pyridoxal phosphate by kynureninase leads to a valuable diagnostic tool. The excretion of xanthronate in response to a tryptophan load is diagnostic of vitamin B6 deficiency. This occurs because the conversion of the tryptophan metabolite, 3-hydroxykynurenine to 3-hydroxythranolate, is impaired due to the lack of sufficient PLP. As a result, a large portion of 3-hydroxykynurenine gets shunted towards the formation of xanthronate, which is then excreted in the urine. It's a very specific metabolic bottleneck that alerts us to a broader nutritional problem, lack of B6. The second notable condition is Hartnup disease. Now, this is not a defect in the catabolic enzymes themselves. Rather, it reflects an impaired intestinal and renal transport of tryptophan and other neutral amino acids.
Another transport issue.
Exactly. Because tryptophan isn't absorbed properly from the diet, intestinal bacteria act on the unabsorbed tryptophan to form various indole derivatives. These are then absorbed and excreted in the urine. Crucially, this defect limits the availability of tryptophan for the body's own niacin biosynthesis. Niacin is vitamin B3.
And niacin can be made from tryptophan.
It can, yes. This accounts for the pellagra-like signs and symptoms observed in patients with Hartnup disease. Niacin deficiency causes pellagra, characterized by the 3Ds, dermatitis, diarrhea, and dementia. It's a prime example of how transport defects can lead to complex and seemingly unrelated metabolic consequences. Highlights the importance of proper nutrient absorption.
Methionine, often referred to as active methionine. What's its catabolic journey like? And how does its breakdown contribute to the body's overall metabolism?
Methionine's catabolism is tightly linked to its crucial role as a donor of methyl groups. It's fundamental. It first reacts with ATP to form S-adenosylmethionine, SAM, which is indeed widely known as active methionine. This designation comes from its very high group transfer potential, particularly for transferring methyl groups in numerous vital biochemical reactions: DNA methylation, neurotransmitter synthesis, creatine synthesis, many others. Subsequent reactions in its catabolic pathway after the methyl group has been donated and SAM is converted to S-adenosylmethionine lead ultimately to the formation of propenyl-CoA. Propenyl-CoA is then converted to succinyl-CoA through a series of well-known reactions that are part of general metabolism. This conversion involves biotin, vitamin B7, and vitamin B12 as cofactors.
So more vitamin connections.
Yes. And this conversion makes methionine's carbon skeleton glycogenic, meaning it can contribute to glucose synthesis via succinyl-CoA entering the TCA cycle. Defects in enzymes like S-adenosyl-homocysteine hydrolase or methionine-adenosyltransferase can lead to the accumulation of methionine, a condition known as hypermethionemia. This highlights the importance of each step in this pathway for proper methionine balance and the overall methylation cycle, which is critical for cellular health.
Lastly, let's turn our attention to the branched chain amino acids. So leucine, leucine, and valine. You hinted earlier that their breakdown has interesting analogies to fatty acid catabolism. What are the similarities, and what makes these amino acids particularly unique in their catabolism?
This is where the metabolic parallels become truly elegant and reveal deep evolutionary conservation, likely. Many of the initial reactions in the catabolism of isoleucine, leucine, and valine are indeed functionally analogous to reactions found in fatty acid catabolism, specifically beta oxidation. This shared enzymatic machinery highlights the efficiency of biological design using similar tools for different jobs. The process for all three branched-chain amino acids begins with transamination, removes their amino group, and forms their respective alkydo acids. The resulting alkydo acids then undergo a critical second step: oxidative decarboxylation and conversion to coenzyme A diesters. This multi-step process is catalyzed by a large and absolutely vital enzyme complex located in the mitochondria, known as the branched-chain keto acid dehydrogenase complex, or BCKDH.
BCKDH. That's a mouthful. Is its structure or function similar to other large enzyme complexes we've encountered in core metabolism, like pyruvate dehydrogenase?
It absolutely is, and this is a key insight for mastering this topic. Its components are functionally identical to those of the pyruvate dehydrogenase complex, PDH.
Which connects glycolysis to the TCA cycle.
Exactly. PDH converts pyruvate into acetyl-CoA, a gateway to the TCA cycle. Like PDH, the branched-chain keto acid dehydrogenase complex consists of several key components. There's E1, which is a thiamine pyrophosphate TPP-dependent branched-chain keto acid decarboxylase. TPP comes from thiamine, vitamin B1. Then there's E2, a dihydrolipoyl transacylase, which contains a lipomide moiety. And finally, E3, a dihydrolipoamide dehydrogenase, which contains FAD, derived from riboflavin vitamin B2.
So many B vitamins involved here, too.
Absolutely. And importantly, just like PDH, its activity is exquisitely regulated. It's controlled by phosphorylation, which inactivates it via a dedicated BCKDH kinase, and by dephosphorylation, which activates it via a BCKDH phosphatase. This fine-tuned control ensures that branched-chain amino acid breakdown is precisely coordinated with the cell's energy needs and metabolic status. Following this initial complex, the dehydrogenation of the resulting coenzyme A diesters proceeds much like the dehydrogenation of lipid-derived fatty acyl-CoA diesters, further cementing that analogy to fatty acid catabolism. The subsequent reactions are then unique for each amino acid skeleton. Leucine, isoleucine volley diverges here, leading to various end products like acetyl-CoA, propionyl-CoA, and succinyl-CoA.
Given the complexity of that branched-chain keto acid dehydrogenase complex and its vital role, I imagine what happens when this intricate system breaks down can be quite severe. What are the significant metabolic disorders associated with branched-chain amino acid catabolism?
There are indeed several significant and often severe metabolic disorders that arise from defects in these pathways, underscoring their critical nature. The most well-known is maple syrup urine disease, MSUD, also colloquially called branched-chain ketonuria.
Right. The name is unforgettable.
As the name vividly implies, the urine of affected individuals has a distinctive odor, often reminiscent of maple syrup or burnt sugar. The core biochemical defect in MSUD is in the keto acid decarboxylase complex, the BCKDH complex we just discussed, specifically at reaction 2, the oxidative decarboxylation step. This defect leads to significantly elevated plasma and urinary levels of leucine, isoleucine, and valine, as well as their cognate keto acids and hydroxy acids. It's important to note that while all three branched-chain amino acids and their keto acids accumulate, the urinary keto acids derive principally from leucine. Leucine and its keto acid, ketosocapirate, are thought to be the main contributors to the characteristic odor and, more importantly, the severe neurotoxicity seen in MSUD.
What are the clinical signs and symptoms of MSUD, and how is it managed? It sounds like an urgent condition.
The signs and symptoms of classic MSUD typically appear in the first few days of life. They often include severe ketoacidosis, significant neurological derangements, lethargy, poor feeding, vomiting, seizures, and profound intellectual disability if untreated. The exact mechanism of toxicity from the accumulating compounds isn't fully understood, but the consequences are devastating. Early diagnosis, often through newborn screening detecting elevated leucine levels followed by confirmation via enzymatic analysis, is absolutely essential to avoid irreversible brain damage and early mortality. Treatment primarily involves replacing dietary protein with a specialized medical food, an amino acid mixture that specifically lacks leucine, isoleucine, and valine.
A very strict diet for life.
Yes, exactly. A lifelong, rigorously managed diet is required to prevent the buildup of these toxic compounds. The molecular genetics of MSUD are quite heterogeneous, meaning different genetic mutations can cause the disease. MSUD can result from mutations in the genes encoding any of the complex's main components, E1, E2, or E3. This leads to recognized genetic subtypes, type IA, type IB, type 2, and type 3, respectively, which can sometimes have varying severities or responsiveness to certain treatments, like thiamine supplementation in some E3 deficient cases. Another variant is intermittent branch-chain ketonuria. In this form, the BCKDH complex retains some residual activity. It's not completely deficient. As a result, symptoms tend to occur later in life, maybe in childhood or even adulthood, and they are often triggered episodically by stressful events, like infection or surgery, or by periods of very high protein intake, which overwhelms the limited enzyme capacity. We also have isovaleric acidemia. Here, the impaired enzyme is isovaleril-CoA dehydrogenase. This acts further down the leucine catabolic pathway after the BCKDH step. Upon ingestion of protein-rich foods, this defect leads to elevated levels of isovalerate, which is the deacylation product of isovaleril-CoA. Clinical symptoms include episodes of vomiting, lethargy, metabolic acidosis, and potentially coma. The accumulated isovaleril-CoA is hydrolyzed to isovalerate and then excreted. This gives a very characteristic sweaty feet odor to the urine and sometimes the breath, a distinct diagnostic clue. Our sources also mention hypervalinemia, which is a defect in valine transamination, the very first step for valine, and methylmalonic aciduria. This is a defect in the conversion of propionyl-CoA, derived from isoleucine, valine, methionine, threonine, and odd-chain fatty acids, to succinsyl-CoA. So methylmalonic aciduria affects multiple pathways, not just branched-chain amino acids. It involves vitamin B12 metabolism as well, though the sources don't detail their mechanisms here.
So having delved into such incredible detail, exploring all these pathways, enzymes, cofactors, and devastating diseases, what does this all mean for us, the listener? Beyond the intricate biochemical diagrams and complex enzyme names, what's the overarching takeaway for someone who wants to grasp this like an expert, as we set out to do?
Well, I think what we've explored today really highlights the incredible complexity, but also the absolute precision required for your body's metabolic machinery to function correctly, day in, day out. The core insight is that each catabolic pathway, each enzyme, each coenzyme, it's not just a theoretical concept in a textbook. It's a critical, indispensable component in converting the food you eat, specifically the amino acids from proteins, into the exact energy you need or the precise new building blocks for your own tissues, carbohydrates, and lipids. It's really a testament to the elegant efficiency and interconnectedness of biological systems, a finely tuned orchestration happening continuously, mostly without us even noticing.
And when even one tiny piece of that machinery is defective due to a genetic mutation, the consequences can be profound, sometimes devastating, as we saw. Conditions ranging from PKU, where a single enzyme defect can lead to severe intellectual disability if untreated, to maple syrup urine disease, causing profound neurological issues in those distinct odors. But the power of modern diagnostic tools like tandem mass spectrometry and newborn screening is truly life-saving. Detecting these rare conditions early allows for interventions, like those highly tailored dietary restrictions that can prevent severe brain damage and early mortality. It's a remarkable fusion of deep biochemical understanding and practical medical intervention.
Absolutely. And the historical insights, like the remarkable discovery of L-Captonuria in that ancient Egyptian mummy, and its foundational role in Sir Archibald Garrett's early 20th century ideas about inherited metabolic disorders. They remind us that while our diagnostic tools and molecular understanding are relatively new, these fundamental biological questions about human health and disease have been with humanity for a very, very long time. It also offers a glimpse into the future. The ongoing research into advanced therapies, like genetic engineering or enzyme replacement therapy, offers a tantalizing glimpse into a future where permanent corrections might be possible, moving beyond managing symptoms to potentially curing the underlying defect entirely for some of these conditions.
So, as you reflect on this deep dive into the hidden yet profoundly vital world of amino acid metabolism, consider this provocative thought. How might our growing, intricate understanding of these specific metabolic pathways and the precise genetic defects that impact them, how might this fundamentally change how we approach personalized nutrition and precision medicine in the coming decades? What new frontiers might be unlocked when we can truly tailor interventions, dietary recommendations, maybe even preventative strategies to an individual's unique metabolic blueprint, potentially preventing disease before it even fully manifests or even correcting it at its root? It's a fascinating future to consider. That's all for this deep dive. We hope you've gained a clearer, more profound understanding of the fascinating world of amino acid catabolism and the remarkable science behind diagnosing and managing these inborn errors of metabolism. It's a topic that truly underpins so much of human health and biochemistry.
Knowledge is most valuable when it's understood and applied. Keep asking those questions. Keep seeking those connections between different pathways. And remember, there's always more to learn about the intricate and beautiful machinery of life.
Stay curious, everyone, and we'll see you on the next deep dive.